All right, so this week we're going to start talking about cell division. And the reason we talked about chromosomes and we talked about DNA and DNA replication is because those things are an important part of the process of cell division. Now, there's two types of cell division. One type happens in simple organisms, and another type happens in more complex organisms. So the first type is binary fission. And binary fission is going to happen in simple organisms that only have one copy of DNA, and it's basically a copy DNA divide. It's not difficult at all. The other types are mitosis and meiosis. We're going to just focus on mitosis right now, but that is a much more complex division process because the organisms have more complex organelles. So binary fission is found in prokaryotes. Prokaryotes are single-celled and have no nucleus, remember. So when you have binary fission, it is a copy the DNA and then split the cell in half, and the cells are identical to each other. This is much faster. So like in something like a bacteria, this could take like only 30 minutes, whereas it could take hours to days in a more complex organism. So some of the differences are in binary fission, you're going to have one haploid, and haploid means half the number of chromosomes or one set of chromosomes being copied. It results in two identical haploid cells, and it's only going to happen in prokaryotes. And then in mitosis, it's a diploid cell being copied, and diploid means two copies of DNA, and it results in two identical diploid cells, and it's going to occur in eukaryotic organisms. We're going to later talk about meiosis, which is a little bit more complex, but we'll talk about that when we get to genetics. So here's what it looks like. Mitosis, you're going to have to like copy everything that's in the nucleus, and then you got to line everything up, the chromosomes like we talked about. We're going to talk about these steps much more in detail during this video and next week. And then binary fission is simple. Copy, copy, split. So the thing we want to know is like, how do we get from this to this to this? How does that happen? It all begins with, we've talked about this a couple times, the fertilized egg. So once the sperm fertilizes the egg, that zygote goes through cell division, like repeatedly, over and over and over and over and over again, through mitosis to give rise to identical cells. Once that zygote reaches a certain size, that differentiation occurs, that process that creates special structures and functions and gives cells a job. Side note, there's not a section for this on your note sheet. It's okay. Write it on a separate sheet of paper, jot it down somewhere else, or just come back to this part of the video later. So the specialized cells are eventually going to become tissues that have a specific job, organs, and organ systems. All right. So that fertilized egg is going to grow, all right, here, and eventually it's going to turn into this more complex organism that starts to have specialized cells. Those cells that are first formed in the zygote are what we call stem cells. They're undifferentiated cells that can become many different types of cells, okay? So it can turn into a nerve cell, or a digestive cell, or a liver cell, a cardiac cell, or a bone cell. There's other types too. So an embryonic stem cell are cells that have never differentiated before. So they're like brand new cells. They start dividing and they turn into these new cells. Adult stem cells can be found in bone marrow and are partially differentiated. So we talked about this. There are in the axolotls, the blastema cells, right? They can become certain things. Adults have that too in bone marrow, but they can't go backwards like they can in the axolotls. The adult stem cells can be found in bone marrow, but they're partially differentiated and they can only turn into bone, blood, cartilage, fat, and connective tissue. So this is important because these turn into what we call gastrula, which are an embryo with 3 different cell layers, meaning they can become one of these 3 things. And over time, that process turns into the development of organs and organ systems. So this is a complex process, Okay. And remember, we start with organisms, right? To organ systems, organs, tissues, cells. So that's how we're going in that order, right? Organism, then we move down to organ systems. So we are going to get all these different types of organization in living things. Now, the cell cycle is a repeating set of events that make up the life of a cell. Cell division is only part of the cell cycle. There is this time between divisions that we also have to talk about. So the cell cycle is mitosis and cytokinesis, which is the division of the nucleus and the cytoplasm. Interphase is all the time in between. And if you see this picture over here, this big section is interphase, and that small little sliver, that's mitosis. So it's much less time. So interphase is the process of the cells becoming ready to divide. So there's three parts. There's G1 or gap 1, where the new cells are growing. They're growing to reach a specific size. Then you get to the synthesis phase, right? I like to always think about these as terms of like life, parts of your life, like life cycles. So G1 is like baby to, 11 or 12. All right. You're just growing. You're getting up to close to adult size. In synthesis, after the cells have reached mature size, they replicate the DNA. So think about G1 as, if you go through puberty early, it's 11. But let's say it's ages 1 through 15. That's growth. You're almost adult size. In the synthesis phase, you're technically ready to reproduce. Not actually, don't start reproducing yet. You're not really ready. But your cells start copying the DNA through DNA replication, like we talked about. That's the S phase. So remember, synthesis means to make. So here we're making DNA. And the G2 phase, that's when we divide the organelles and prepare for mitosis. This is when we're like out of college and we're ready to start our life. We are actually getting things ready. We're buying a house and we're having a job and we're saving money. This is the G2 phase. It's the second growth phase. So G1 is first. We're growing. G2, we're copying our DNA, getting ready to reproduce. S phase is copying the DNA and getting ready to reproduce. G2 is growing again and waiting for those final stages of cell division. Mitosis occurs in 4 phases: prophase, metaphase, anaphase, and telophase. We often refer to it as PMAT. Mitosis is the division of the nucleus, okay? Just the nucleus. The first phase of mitosis is called prophase. Prophase, you have DNA coiling and shortening. The chromosomes are going to attach together at the centromere. We start seeing centrioles forming, and these are spindle fibers. In prophase, we're packing. We're filling up our suitcases, getting ready to move, okay? Prophase. So here's what prophase looks like. The chromosomes are condensing. All of these things are happening during that prophase. The next is metaphase. It's the second phase of mitosis. In metaphase, the cells line the chromosomes up in the middle of the cell. They're easier to identify. And then the spindle fibers start attaching to these chromosomes at the centromere, but they're all lined up in the middle. So metaphase is in the middle. Here's the steps again. So metaphase, the chromosomes are aligning in the middle, and we've already watched the steps of prophase. In anaphase, that's the third phase of mitosis. The chromatids, so the original and the copy are attached together, remember, they're going to pull apart. Okay, so anaphase stands for apart. They're going to move towards the poles of the cell, getting pulled at the centromere. All right, so here we see that happening in anaphase. They're starting to get pulled apart. I don't know why it's not happening yet. There we go, pulling apart. And then the final phase is telophase. It's the final phase of mitosis. The chromosomes are at the opposite end. They start to uncoil, making our spaghetti DNA, our chromatin, and the nucleus begins to form in the new cells. So telophase, think of 2, all right? Or telephone poles, chromosomes are at the poles. So all of these things are considered telophase. All right, I sped through the end because my video was ending, so I'm pushing 2 videos together. So Telophase is this final phase of mitosis where now you have two copies of your DNA. You have your nucleus starting to reform here. The nucleolus, that's this little purple dot, is starting to reform. You have basically the start of two new cells. So if you take a look at this process, here's your chromosomes getting pulled apart. In telophase, they're at the very end here. And then here's your two cells that you're sort of looking at, your new cells. The last part is called cytokinesis. And in cytokinesis, it's sometimes considered a part of telophase, but it's the division of the cytoplasm. So in animal cells are going to pinch in and pull apart. They're going to pinch in and pull apart at what we call the cleavage furrow. And it kind of looks like a butt, right? But it's going to pinch in and pull apart. That's different than in plants. In plants, they form what we call a cell plate. So they have two new cells, and then a plate starts to form in between the two. Okay, so a little cell plate forms, and that becomes the cell wall. All right. So cytokinesis is just dividing the cytoplasm. The nucleus has already been divided, and now the cytokinesis part is just dividing the cytoplasm. So that's what you see here. In this final step, you have two brand new cells, and the cell is finally dividing into two new identical cells from the original. Let's watch this short clip here.
The purpose of the cell cycle is to reproduce cells. So, one cell can copy itself to become two cells. Those two daughter cells are identical to the original parent cell, exactly the same. Each one can complete its own cell cycle to make copies of itself until you end up with lots of cells. This is needed for growth and is how you went from being one single lonely cell to being made of trillions of cells. That's how you grew. We also need the cell cycle to replace worn out cells and heal tissue damage. So it is muy importante. All right, so the cell.
All right, that's a 5-minute video. I'm going to link it for you in the directions of this video so that you can add it and watch it if you wish. It's long. I already talked for 5 minutes, more than that. So I don't want to spend any more time, but you can watch it on your own if you like. This is what an actual cell looks like during interphase, and this is like an artist drawing. This is interphase, the chromosomes. You got that chromatin. The chromosomes aren't formed yet. You start to see the chromosomes forming a little bit here, and then you start seeing them really forming here in prophase. Here's metaphase. All the chromosomes are lined up in the middle. Anaphase, they're pulling apart. And here is telophase, where the chromosomes are centralized at the poles of the two new cells. Okay, there is like 900 bazillion different images you can look at mitosis. These are all accurate. So take a minute here. Which cell do you think is in prophase, metaphase, anaphase, and telophase? A lot of this is going to be just memorization of what the pictures look like. We're going to do some activities where we look at cells. We're going to do it online. We're going to do it with the microscopes. We're going to make drawings. So you're going to have lots of opportunities to practice looking at these cells. Okay, so this first one is prophase. Then you have anaphase. Then you have metaphase. And then the last one is then obviously telophase. Okay, so make sure that you understand the steps and also the order. Remember, it's always going to be PMAT: prophase, metaphase, anaphase, telophase. We're going to talk about cell regulation in a different video. I hope you guys have a great weekend. I hope it snows a lot for us. Knock on wood. Have a great day.